Alright guys, welcome back to Underground Science, and in this video we're going to be talking about telomeres and telomerase. Alright, so this is only in eukaryotes with telomerase, and so basically what we call this is the end replication problem, because it's a problem when you end replication, okay? So here's what happens, right? So remember from our previous video in DNA replication, what did we have? We had a leading strand, okay, which is a strand that's 3' to 5', and then we had the lagging strand, which was 5' to 3'. Remember, the leading strand did not have Okazaki fragments because we only used one primer, which we synthesized from 5 to 3, and then DNA polymerase 3 came in and started on the 3' end. Remember, this is going to be very important when we talk about telomerase, okay? DNA polymerase 3, right, needs the 3' end of the primer in order to synthesize the rest of the nucleotides, DNA nucleotides. Okay, so it needs a 3' hydroxyl end in order to synthesize the rest of the nucleotides. So DNA polymerase 3 will come in, and then I'm not going to draw the nucleotides, but I'm going to erase this 3' end because DNA polymerase 3 will come in, and then in green will show DNA nucleotide synthesis. Okay, so it's going to replicate and create a complementary strand. It's going to replicate that way because it's 5' to 3', right? Same on the lagging strand though. If we look at the same thing on the lagging strand, which is right here, right? Let's scroll down just a tad bit. On the lagging strand, remember, we're still doing the same thing. You're still synthesizing from 5 to 3. But the only problem is DNA replication's going that way, okay, to the right. So you're synthesizing your DNA nucleotides on the lagging strand in the opposite direction. So let's say we put a primer here, 5 to 3, right, anti-parallel, because this is 5 to 3. So we synthesize the RNA primer from 5 to 3 this way. And then DNA polymerase 3 comes in, right, represented with the green arrow, okay, and synthesizes the DNA from the 3' end of the primer and on, okay? So it's synthesizing from 5 to 3, right? Because this side, when you're going left, this side is 5 to 3 if you're synthesizing, because it's anti-parallel fashion, right? DNA replication, DNA is anti-parallel. So if you're 5 to 3 here, right, and you're synthesizing this way, then you're synthesizing in anti-parallel fashion, means you're synthesizing from 5 to 3, okay? Main thing we have to focus on, again, you see this primer, right? The 3' end of the primer is what the DNA polymerase 3 needs in order to synthesize the DNA. So again, we come to this primer. DNA polymerase 3 locates the 3' end, because that's the end that has the OH group, and then the DNA polymerase 3 synthesizes the DNA, right? Again, locates the 3' end, DNA polymerase 3 does, then synthesizes the DNA in the 5 to 3' direction. Again, it comes here now, right? It comes here now, right? And then it would, if we had more nucleotides, that would just keep on happening, right? Then remember what happens. DNA polymerase 1 comes in, okay, and it takes out the RNA primer. Okay, so let's do that now. So what we're going to do is, let's start here. DNA polymerase 1 comes in with its 5' to 3' exonuclease activity that we talked about in the previous video. DNA polymerase comes with its 5' to 3' exonuclease activity and takes out the RNA primer from the 5 to 3' end, right? Then we add the DNA nucleotides, which we'll represent with green, okay? DNA polymerase 1 will do that from the 5' to 3' direction, okay? So we're going to have C and A. Cool. DNA polymerase 1 will again go here with its 5' to 3' exonuclease activity. It will take this primer out, okay? And then it will synthesize the DNA nucleotides that's perfectly complement to the 5' to 3' strand, which will be, so if we draw it like this and here, that's going to be guanine, cytosine. Okay, right? So, again, DNA polymerase 1 will come here, right? Take this primer out, right? Take this primer out, okay? Then it'll try to look for the 3' end, because remember, DNA polymerase 1 and 3 both need, in this case we're talking about DNA polymerase 1. DNA polymerase 1 will look for the 3' end because from the 3' end is is where it will synthesize the DNA nucleotides. Okay? This part, okay, of the DNA, this part, this arrow represents the DNA that's been formed, right? This part right here, so if we go with red, right here, this right here, this is the 5' end, okay? Because this is from 3 to 5' that way, so it's going to be from 5 to 3 this way, okay? There is no 3' end anymore. So DNA polymerase 1, when it reaches the end, hence end replication problem, it cannot locate a 3' end in which to build the DNA, right? So what happens is, so like for example, right here, let's go back. Let's say we're on this primer. This was a primer before, right? Let's say DNA polymerase 1 took this out, okay? Real quick, right? DNA polymerase 1 took this out. Then, so took out the primer. Now, it'll try to locate the 3' end. Where's the 3' end? Okay, right now, at this moment, when it took out the primer. So remember, 3' end is right here, right? Because this is going to be the 5' end, so then this is going to be the 3' end. So then DNA polymerase 1 will synthesize the DNA from here to here, starting from the 3' end and on, right? Same thing happens here. When you take out the primer, which was over here, it tries to locate the 3' end, but there is no more DNA here, right? Like, because it's the end of the sequence, it's the end of the strand. So you can't locate a 3' end, therefore you cannot lay any more nucleotides down. So what happens is, this strand that you just formed on the top, so if we erase all this, right? Erase all this because we don't need this right now. Okay, so this strand that you formed on the top, right? Complement to this strand. So if we drew this right here, in like, since there's a lot of stuff here, what we're just going to do is draw. So remember, right? Remember what happened actually before this. Let's undo this. So remember, we can't form any nucleotides here anymore. So the strand that we just replicated is actually shorter than the original parent strand, right? So if we just highlight this whole thing, right? This is this new DNA strand from here to here. When we took out the primer here, there was no 3' end where DNA polymerase 1 could locate, so we could not replicate these two, right? These two nucleotides at the end of the DNA. Okay, so the strand is now shortened. It's from here to here. Okay, so if that keeps on happening with each time the cell divides, you're going to keep on shortening the end of your chromosomes. All right, and that's why you have telomeres at the end of your chromosomes, which help basically it helps protect you from, from involving your genes, okay, your actual genes of your DNA. Because you don't want to start destroying the genes of your DNA or shortening the genes of your DNA that actually code for proteins. Okay, so at the end, what happens is telomerase comes and actually basically lays down telomeres. Okay, that's why it's called telomerase. So here's what's going to happen, right? Let's say we reach the end. Okay, let's say we have reached the end where on this side, right, let's say we have a telomere. Okay, so let's say we have a telomere on this side, okay? And with each cell division, okay, let's say this telomere has shortened and shortened, just like this strand shortens, because when this strand goes to replicate, right, then we'll shorten again, and it'll shorten again, and it'll keep on shortening over and over, right, because this problem will keep on happening. So what telomerase does is it comes to the end, this 3' end, okay, and it's going to have an RNA template with it, like telomerase will have the RNA template, okay? Because the RNA template that it has, if we go just a little bit down, the RNA template it has is A, U, C, 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 okay? That's the RNA template that is in telomerase. Like telomerase has that template with it. So and then what it's going to do is it's going to take this RNA template and then synthesize DNA onto this 3' end, okay, on this 3' end. So because you're taking an RNA template and making DNA, telomerase is also known as a RNA dependent, right? Because we're dependent on this AAUCCC template, which is RNA. RNA dependent DNA polymerase, because we're synthesizing DNA from RNA. Okay? We're synthesizing DNA from the RNA template. So this RNA template, right, if we convert this to DNA, it's going to be TTA-GGG, right? So TTA-GGG. So let's go and write that down. So telomerase will come to the 3' end. Okay, so let's erase this. And then lay down a bunch of these, okay? Crap, sorry, I didn't mean to do that. Lay down a bunch of these templates of AAUCCC, and we'll lay down a lot of these DNA, complement DNA nucleotides, so that we elongate the end of the DNA. Okay, so let's go and show how that happens. So it's going to come to 3' end. Telomerase will come here, and then we're going to have, let's say we add 1, 2, 3, 1, 2, 3, and then we go even more, add 6 more. Okay, so again, this is a 3' end, right? What it's going to do, telomerase will present this template over here, and then it'll convert this RNA to DNA. So what is that going to be? So we're going to have TTA, GGG. And then telomerase will, so remember, telomerase came over here, right? Let's say this is telomerase, and it laid down this DNA nucleotides from this RNA template that we saw. Then telomerase will come over here now and do the same thing. It'll present the RNA template, and then from here we're going to lay down again, right? This will happen many times, but we're just showing it twice. Again, we're going to have TTA, GGG. Then, right, so now we have elongated the end. So we just prevented, we're about to prevent, right? So what's going to happen is, now what you can do is an RNA primer can come over here. Okay, so remember this is the 3' end. So an RNA primer will come in, and let's say the primer lays its the primase will come. Remember, that's the enzyme that lays down the RNA primer. So if we drew that, the primase can lay down the primer right here, right? Let's say this primer is 2 nucleotides long, it's RNA. So this would be CC. And then DNA polymerase can come in and lay down nucleotides, DNA nucleotides from here to here, right? So look what we just did, right? We just elongated this strand, the end of this strand. So now we have a lot more nucleotides at the end, right, called telomeres, right? These are our telomere sequences, TTA, GGG. We have a lot more telomeres, a lot more nucleotides to sacrifice now when this cell undergoes more divisions, rather than like reaching a point where, let's say this point and on was genes, right, that would code for proteins. Like, you don't want to keep on shortening your DNA until you get to the genes that code for proteins, because then that's, it's going to be bad. You won't be able to have proper functioning proteins then if you start deleting the nucleotides that are involved in genes that code for proteins. So see, what we did is this strand got way longer now because we added more telomere sequences like TTA, GGG, and then we replicated the other strand based off the telomere sequences, TTA, GGG, right? And so that's basically what telomerase does. It's actually pretty cool because if you imagine, right, if the more telomeres you have, the more telomerase activity you have, right, the better your outcome will be, especially because telomerase is involved in aging. Okay, so we should note that down. That's something important. Telomerase is involved in aging, right? Because the more telomerase you have, the more your cells can replicate. Okay, because the more telomerase will put on more telomeres, which will mean you have more DNA at the end of your chromosome to sacrifice with each cell division. So your cells can divide more before it basically can't divide anymore, right? The Hayflick limit. So that's if we're talking about the fact that cells that replicate a lot, right, need telomerase to function. So that means it would make sense to say that cancer cells, right, can upregulate their telomerase. So you have a lot of telomerase activity in cancer cells. That's why cancer cells, they proliferate and replicate way too rapidly because, one thing that can help is high telomerase activity, which means, they can divide way more times, like a higher number of times before they can't divide anymore, because more telomerase means more telomeres, means more DNA to sacrifice with each cell division before those cancer cells, reach nucleotides involved in their genes, So that's where cancer cells, so that's one example, I should say, right, for cancer cells, for telomerase activity, which is in cancer cells. It's also involved in aging, things like oxidants, smoking, all that stuff, right? That's bad for your telomeres. You can also, like antioxidants, for example, are good for your telomeres. So, just things like that. It's pretty cool if you relate this to, real life clinical significance. So that's basically telomeres and telomerase. Um, the main thing that we need to know is that it basically elongates the end of your DNA, right? When your replication's ending, at the end, this strand will shorten, right? Because DNA polymerase 1 does not have a 3' end of the primer, or when it takes out the primer at the end, it cannot locate a 3' end. So therefore, it cannot synthesize new DNA. So your DNA strand, every time you replicate, your end of the DNA, end of your chromosomes, basically keep on shortening. And you have telomerase to add telomeres to elongate that strand, so you don't keep on shortening your DNA until you hit the genes that actually code for proteins. All right, so that's basically it for telomeres and telomerase. All right, there's probably explained it too much, but hopefully it made sense at the end of the day. And I hope I see you guys in my future videos. All right, yeah, thanks for watching.